Welcome back. In this module, we're going to talk about understanding different types of experimental research designs. Let me reemphasize that for most of the IRL teams, we will not be doing experiments, and certainly not sophisticated experiments. But it is my hope that by sharing and coming to some common understanding about what a true experiment would look like or could look like in different ways, we can do better research, whether you're doing photo voice, a particular case study, or so many of the other community-based participatory research methods we're all involved in. So again, this is to open our eyes, to extend our thinking in order to do better work in what we're doing. So let's talk about how to understand, if you will, diagram experimental designs. First, just a little bit of language. In the following slides, you'll see an O that represents an observation moment, like September 2016 or whatever else date one would wish. The X represents an intervention or treatment. We changed the policy, we gave more fruits and vegetables to the people, we did this, we did that. That's the X. The R represents that we were able to randomize people to the experimental condition or the control/comparison condition. So the R is randomization, and the NR, N as in Nancy, NR is for not randomized, and that'll be clear. Our very first type of experiment is perhaps the simplest. Notice in this case, what we are doing is just observing people after some intervention, where since both rows have an R in front of them, we have randomized people, hypothetically, to either the experimental condition or the control/comparison condition. So this is sometimes called a post-test only experiment. And it's fascinating and it's very effective. Were we able to randomize enough people, say 1,000, 2,000, to treatment or a control, and just observe them after the treatment, we'd have very good comparison. In fact, that treatment arm, the upper row there, serves as a counterfactual to the lower arm, which is the control arm. So the only difference between these two conditions, where they might represent 1,000 people each or maybe more, is that one group got treated with the X, the other one didn't. So that's the but-for condition that we spoke of earlier in this course. And so we compare treatment to control. To the extent that they're different, we say that difference is due to whatever treatment we applied, the policy change, the menu labeling, the community enhancement, whatever it was. So all the outcomes should be the same, except for the outcomes that were changed by the treatment itself. That's why this experimental design is simple and powerful and very effective. You might ask, however, well, maybe they were different at baseline. They were different before. 2 communities, 5 communities, 600 people, whatever numbers you want to put to it. Maybe they were different before the randomization. The fact is, if we could randomize enough people, 1,000, 10,000, maybe more, that would all be the same. As randomization would take care to make sure that the two groups, treated and control, were the same, what we technically call balanced. They are on average the same. So the same average numbers of males, the same average number of wealthy people, the same average number of tall, short, green, big-footed, whatever feature you want, are all the same, including on stuff we can't measure. That's the power of randomization. If we weren't able to randomize a lot of people, we might want to do a pretest. Well, that design is simple as well. Here, we can imagine, look, an observation before the treatment itself, and then an observation after the treatment. And the same thing in the comparison or control arm. That's the lower sort of row, if you will. Now, again, we're still randomizing, but sometimes we can't randomize enough people or are actually interested in the change from baseline to follow-up. And so here we have more comparisons we can make. Look, we can make that old post-test only comparison, and that works great. We assume the pre-tests are the same. On average, they're all the same. But we can also do a before and after within treatment arm. And for those of you who've worked in this area, this kind of design lends itself naturally to the so-called difference in difference estimator. That as we subtract baseline from follow-up in each arm, and then subtract those two differences. That's all it is. But the important thing is all we're doing here is comparing a group that got something to a group that didn't get something in order to see the causal effect of that thing we applied, that treatment. Again, it's a policy, it's a social norm change, whatever it is we're working on. These kinds of designs can be enhanced by what we call blinding, and sometimes we can run, if possible, a double-blind experiment. In this case, neither the researchers nor the subjects know what the treatment allocation was. So you might hypothetically receive a white pill, and you don't know if that's an active drug or just a sugar pill. That has advantages because then neither the researchers nor the subjects can overly change the results of the study due to their own personal biases. If you know you're being treated, you might say, hey, this thing's working. If the researchers know you're being treated, they might measure you differently. So double-blind or blinding studies is another mechanism to reduce bias and to get closer to the truth of the answer. Now, these double-blind studies are sometimes the gold standard for pharmacological studies. The Food and Drug Administration, or FDA, likes these kinds of studies. But they're really not feasible, if at all, in sort of the social determinants of research or the social research more generally that we're all working on. For a non-blind randomized controlled trial, as recently diagrammed, the researchers and/or subjects know if they've been treated or in the control comparison condition. This is what's common. Now, sometimes we can blind subjects, a community, they don't know if they're receiving an actual cooking class or maybe that's part of the comparison condition. So sometimes that can work, but it's not as pure, if you will, as a drug trial. But the model remains the same. We're trying to understand the impact of the intervention on the outcome measure or measures in order to find what the true impact is as best as possible. We can go yet a step further and talk about something called crossover experiments. In this case, I might receive a placebo at first, and then after some period, receive the actual treatment itself. You might receive the actual treatment itself first, and then the placebo later on. And so we're crossing over. We are both getting everything at just different times. Again, this is a lovely design, rarely feasible in social research, but I want you to understand it because the important thing is everybody is getting everything, and therefore the comparisons can sometimes be more defensible. So whether we can actually apply this kind of work in the IRL framework is not so important. What's important now is understanding what's actually happening here. And we can diagram it very simply as well. Again, 2 rows, 2 treatment conditions, treated and controlled. But in this case, there's treatment A, maybe that's the control, and treatment B, which is the actual active treatment. And each person gets both, probably at different times. And we can randomize that as well. So that's just scaling up or making more sophisticated the very simple post-test only experiment we spoke of a few moments ago. We can go even further, and I want to just present this to you in a simple way to help you understand that there's more going on here. And what's going on in this case is we might have 2 kinds of treatment. We might have changing menus by adding calories and maybe changing education level of people who eat fast food through an education program apart from calories. So here we have 2 kinds of treatment. When we have more than one kind of treatment, we tend to call these factorial experiments, when everybody gets some or part of both kinds of conditions. Now, these kinds of experiments get very sophisticated very fast because we have many levels of treatment quickly. That is, if we have 4 factors, 4 different things we want to do, at 2 levels each, yes or no, we quickly have not 2 treatment conditions, but 16. So lots of comparisons to make, and sometimes that can be challenging analytically to figure out what happened. We can make a simple table to understand what this factorial thing means. Again, just consider 2 treatments, A and B. And it can be anything. It can be helping people, maybe young people, learn how to read better. That's A. And B might be teaching parents about nutrition. And the question is it reading or nutrition that's improving health? So the outcome of the study might be some measure of health. How do you feel? Maybe it's blood pressure, whatever it is. But we have 2 things we're going to change. And so in the first group, we have yes A and yes B. So yes nutrition and yes education. In the second group, it's perhaps no A and yes B, and then all the way down the line. So we get 4 potential conditions here with 2 kinds of interventions at 2 levels each, yes or no. And obviously that gets sophisticated very fast. But the point here is when we have 2 things we think we want to manipulate, it becomes increasingly important to imagine what that ideal experiment will look like if we were to run it as such. Yet another kind of experiment is instead of randomizing individual people, so Jane gets treated, Joe does not, Sally doesn't get treated, Pamela does, on, individuals themselves being assigned to treatment or control. We might assign whole groups, sometimes called clusters, where a whole community gets a new nutrition program and another community does not. Or maybe a whole clinic gets a new way to treat diabetes and another clinic does not. There's all kinds of ways we can imagine clusters, sometimes viewed as families, schools, communities, states, even herds of cattle. All kinds of things become clusters. What's different here is instead of randomizing individual, we randomize the whole cluster, the whole group, to treatment or control. This brings up some more sophisticated analysis, which we'd want to talk about in another time. But what matters here is you can imagine in your mind, I hope, not randomizing particular persons, but whole groups. The big challenge of these cluster randomized trials is they tend to have very low statistical power. That is, it's hard to discern whether the treatment actually worked or it's just chance alone, just random noise. Finally, with respect to experiments, sometimes we talk about something called a natural experiment. Now, this is an experiment just like we've always talked about, an exogenous treatment and randomization to treatment or control conditions. What's different here is instead of researchers, human beings, assigning people at random to treatment or control, Mother Nature does it. So there might be an earthquake that, of course, rattles some community, where another comparison community is not rattled by the earthquake. Now we have a natural experiment of one community being changed and one not. There's also things like lotteries. Military draft is a classic example, where some people get assigned to the military and some not, and what happens to their lives later on. Well, since this is sort of beyond a particular researcher's decision, it's obviously a policy decision, this can be viewed as natural experiments. But the question of policy change itself being a natural experiment, I want to present to you as more of a challenge. Why are policies changed in some school districts or communities and not others? Is that truly exogenous, as we spoke of earlier in the module, or is it because the political power of the citizens or activists in the area? People can compel their policymakers to change decisions. And if that's the case, then the intervention is not exogenous, but endogenous. And as we said earlier, that's a big problem for understanding cause and effect. Related to this whole idea is this other kind of design I just want to lay out for you quickly called regression discontinuity designs. And this is just an interesting way that's kind of observational, but also kind of experimental. It's an interesting hybrid design. What do I mean? Well, sometimes we just call these RDD designs for regression discontinuity. Here, we have a treatment at a pre-specified cutoff. This design was developed in the 1950s, and it's really a powerful, important scientific approach. For me, the best way to understand it is by a simple diagram or graph. Here we can see a graph where you got some sort of vertical line in the center there, right? And then to my left anyway, there's the control condition. And you can see a bunch of dots and then a line through that. To the right of the graph, you see another bunch of dots. Again, we presume those are people, and a line through that. And so what we notice on the horizontal or sort of bottom row axis is there's some assignment variable. And it might be, in this case, the axis goes from 38 to 62, and maybe at level 50. Maybe it's an income thing or an IQ thing, or most importantly, a height thing. And we say we're going to intervene when the score is 50, 5-0. And that's that vertical line in the graph. And so what we see then is that the average score of the individuals, which we mark by the vertical axis, that's ranging in this graph from 34 to 70, is clearly higher after the assignment than before. You can see there's sort of a discontinuity or a jump at that assignment score. And so this is a powerful way to understand what's happening when sort of some policy that has some threshold effect is impacting. So often these things are test scores, they're means tested for income, all kinds of things you can imagine where there's some jump. Well, we can leverage that jump in our research to find out the impact of such changes on the individuals we're interested in studying. This can be expanded, of course, and we can have parallel groups and all kinds of other fancy things. In this case, we see a regression discontinuity design without a change. There's just no impact there at all. And so this is just another way to think about how we get back to that core principle of understanding what would have happened had we not intervened. That's that counterfactual we're seeking. Now again, this can be done whether doing a fancy experiment or just thinking about it if we're doing CBPR or photo voice or something quite qualitative. The principles remain the same, and that's the point of understanding these modules.